Hey everyone, today Jeremiah and I are presenting on Chapter 14, the cell walls. We've broken down our PowerPoint to follow the same format that Professor Baskin used. That is, we're going to talk about composition, assembly, growth, and function. So to start with assembly, it all begins with the phragmoplast. So in the figure in the bottom right corner, you can roughly see the phragmoplast. Now the phragmoplast is a complex of microtubules and endoplasmic reticulum that forms late during cell division. And the role of the phragmoplast is to set up a scaffold for the cell plate assembly, which means that it helps with alignment of everything on the cell plate. So you may or may not know that the cell plate is actually theoretical. It doesn't really exist, but the cell plate is just a point in the cell where everything generally lines up before two daughter cells are formed in cytokinesis. And after division, the phragmoplast lays down the first pieces of the primary cell wall, which orients it for self-assembly. And we'll talk a little bit more about how it does that in just a moment. So here we're going to move on to the primary cell wall. So after the phragmoplast has set up the initial pieces of the cell wall, molecular precursors of the wall have to come together outside of the cell. And those molecular precursors arrive at the cell in two ways, outside of the cell in two ways. The first way is through Golgi-mediated secretion. Following this method, vesicles of the precursors are transported out of the plasma membrane and they're fully assembled in the extracellular matrix. So for example, precursors of hemicellulose might be transported in vesicles out of the cell, and then their last pieces are put together once they're outside of the cell. The other method is through the cellulose synthase complex. And this is more related to how the phragmoplast lays down the initial pieces, where the cellulose synthase complex is an enzymatic complex that constructs microfibrils out of cellulose. And it does this by laying down the microfibrils in the same direction as microtubules at the interior of the cell that were aligned by the phragmoplast. So the microfibrils and the microtubules all line up in parallel with each other when they're laid down from the cellulose synthase complex. Moving on, let's talk for just a moment about what the primary cell wall contains. So it's a hydrated matrix, which means it's a water-based matrix. Primary cell wall and the secondary cell wall too are plastic. And they're plastic meaning that components can change over time and the frequency of components can change over time. The primary three components, which Jeremiah will discuss more in a couple of minutes, are cellulose microfibrils, hemicellulose, and pectins. Moving on to the secondary cell wall, we'll start with a figure here. And the secondary cell wall, as we can see in the figure, is organized and it's built in layers. So here we have one layer, here we have another layer, and this is a third layer of the secondary cell wall. Now we can see the cellulose microfibers here. In each layer, the organization is different, but together, those different organizations help make the overall cell wall stronger. Second characteristic of the secondary cell wall we'll cover is that it forms after primary wall growth stops. So once the cell is done enlarging, then the secondary wall starts to build because there's no longer an effective trigger pressure expanding the wall as trigger overcomes tension in the wall. There's no longer an effect of growth. The next point is that the secondary cell wall is primarily made of cellulose and hemicellulose, which add to the tensile strength of the wall. Fourth, the microfibrils align to form macrofibrils. Now this phenomenon even further increases the strength of the secondary cell wall because if the microfibrils are akin to sticks, then the macrofibrils are like branches. And this further increases the wall strength because all these macrofibrils are then lined up and they're connected by hemicelluloses perpendicular to the macrofibril growth direction, and that's what makes the cell wall strong. The secondary cell wall is also lignified, and it being lignified means that the protein lignin has been added, and that's primarily in the cuticle layer of the secondary cell wall. That helps the wall be hydrophobic or resistant to water intake. The next layer we're going to cover is the middle lamella. So the middle lamella is a very thin, glue-like layer of the size in between cell walls of neighboring cells. And it connects these walls together. And it does so with its glue-like characteristic that keeps the walls from compressing too. So that's another characteristic of the middle lamella. And it gains that characteristic from being pectin-rich. So if two neighboring cells are being pushed against each other, the middle lamella will resist compression of the two cells because it's present in between the walls. Let's move on to functions now. So as we've already discussed, the cell wall has lots of structural strength, and that comes from the microfibrils that run along the length of the cell wall and the hemicelluloses that connect them. The structural strength is important for the plant growing upright to resist the force of gravity. Next, we have prevention of cell motility. So there's no cells slipping and sliding past each other because the cell walls keep the internal cells rigid and fixed in one place throughout the life of the plant. Next, we have sealing gaps between cells. And this is, again, the role of the middle lamella, where all space between the cells are filled in. This helps create a permanent or fixed barrier for plant cells. And of course, we have formation of the apoplast, which we talked about in roots with xylems and with the phloem. So the apoplast is a channel cut through the cell wall, if you will, where nutrients can be supplied to other parts of the plant. And we have regulation. So the cell wall helps with regulation of cell division by limiting the size of the cell. The cell has to grow in order to divide, so the amount of time that it takes to grow is limited by the cell wall because the cell wall is resisting turgor pressure that tries to make the cell grow from water intake. There's also regulation of cell shape because whatever the shape of the cell wall is, the cell inside the plasma membrane will conform to that shape. And there's regulation of turgor pressure too, because as water is intaken into the cell, or there's an influx, the cell wall is also pushing back against the plasma membrane, which helps the cell keep its shape. Next function we'll talk about is the role of the cell wall as a barrier. So invading pathogens such as a bacteria or a virus or molecules that don't belong inside of the plant will encounter the cell wall, but they won't be able to enter the plant because the cell wall forms a continuous barrier around the cells inside of the walls, and that helps protect the plant too. Last function we'll talk about is the cuticle again, which is that lignified layer of the secondary cell wall that helps to resist water loss and it helps to resist water coming in from the outside. So the cuticle, which if you touch a plant leaf, for example, you can very gently remove a cuticle and you'll see that it's a waxy layer that's hydrophobic.
All right. So now we have the cell wall composition. And basically, like mentioned before, the main components of the cell wall are made up of cellulose, hemicellulose, pectins, and other proteins. Now, cellulose is what makes up most of the composition in both the primary and the secondary walls. And it's basically an association of hydrogen bonds, what makes up these microfibrils, which essentially makes up the cell walls. And the hydrogen bonds within the cellulose is what gives rigidity to the cell wall and essentially makes the cell wall strong and be able to stand upright. Okay, next we have, now we have the composition of pectins, and these are water-soluble carbohydrate substances found in the cell wall. And they are basically grouped by polysaccharides that give high extractability, and this allows for the cell wall to move within shape when deemed necessary. And these polysaccharides include xylogalacturins, homogalacturinins, and ramonogalacturins 1 and 2. These elements are what basically provides the backbone for the cell wall, and they are able to keep the cell wall together by linking the adjacent walls of the cell together. Next, we have hemicellulose, and These are basically polysaccharides composed of 2 carbon sugars, D-xylose and 1 arabinose, and 3 6-carbon sugars. These molecules are branched together within the cell wall and are hydrophilic, giving it the ability to hydrate and form gels that basically contribute to the expansion of the cell wall. And they are found in both primary and secondary wall in high abundance. Next, now this is more of a visualization to sort of show you guys where both, cellulose, microcellulose, hemicellulose, soluble proteins are within the primary and secondary wall. There you have your middle lamella at the top. And here we have another more detailed visualization of the primary and secondary wall. And you could see, the pectins are formed through the homoglycerins, the two RG1 and RG2, while hemicellulose is through xyloglycans mixed with B-glucans. And you kind of see how each of these come together to make up the composition of the cell wall. So next we have the growth of the cell wall. And like all things within cells, growth starts with the genetic makeup. And this foundation of the makeup is basically what makes the cellulose, hemicellulose, and the pectins. And they are formed through the synthesis of the Caesar genes. And those genes are both specific and primary secondary walls. And the primary walls, these genes are Caesar genes 1, 3, and 6, while the secondary wall are made up of Caesar genes 4, 7, and 8. And the microfibers of each cellulose within the cell wall come from the bundling of the beta-glucan chains, which stems from these Caesar genes mentioned above. Next, we have more of a detailed of how xyloglucans play a role in cell growth, and they're basically a type of hemicellulose which are key in cell wall growth, and that they're responsible for linking the microfibrils of the primary cell wall together. Now, this linkage is what gives the potential of the cell wall to loosen and the ability to enlarge itself, and the cell wall is doing these motions based on the situation, and it can also vary from plant to plant. Also important to note are that trapped xyloglucans form non-crystalline regions of the cellulose, and once these are released, that results in more cell growth and the extension of the cell wall. Next, we're going to have the microfibrils are formed, and these enlargements are initiated through expansion. And this is responsible for cell wall enlargement because of its ability to influence the cell wall and to stress relax. And stress relaxation is important because it allows for the cell wall to take in water and to actually physically become larger. Next, we are going to see here this expansons, and these are very important because they basically are versatile in how they influence cell wall growth. We have 2 types of expansons. One are exogenous, which are responsible for the stimulus of the cell wall growth, and there are also endogenous expansons, which are responsible for limiting cell growth. And these changes of cell wall growth are controlled by changes of the pH, also known as acid growth. And acid growth basically refers to the ability of a cell and plant cell to elongate and expand quickly. And this expansion is all dependent, once again, on the wall loosening proteins and essentially what type of plant this is. Next, this is a visualization. So here in the before image, you see that this is a smaller cell wall. And then it goes through the process of auxins going through a hydrogen pump through the cytoplasm. And once that enters the cell wall, we see that the hydrogen activates the expansons mentioned before, and those expansons are able to then loosen the cellulose microfibrils, which then elongate water to enter the cell wall and to then expand and essentially make the cell wall larger.